Hello, everybody, and welcome back to Creation Myths. What I'm going to show you here is a conversation I had on Praise I Am's channel way back in March of 2023. This was a small part, about an hour long, of a very long stream. I think it was over like 6 hours long. I just happened to pop in and see that they were talking about the human chromosome 2 fusion. So I popped in intending to just like clarify an argument for just like a couple of minutes, and I ended up staying and chatting for about an hour. So we talked about chromosome 2 stuff and some related topics. So I'm just going to show you that conversation here. There's not much more to introduce, so I hope you enjoy this conversation. I always meant to put it up on the channel, but I just, like, for some reason never got around to it. So here it is, my conversation, about an hour long, on chromosome 2 and some related topics. Enjoy.
Okay, cool. So we gotta argue, base some information on chromosome 2. So is this the claim that they're making? Is what I'm assuming. The chromosome 2 fusion is based on the
Okay,
apes
Dave, I'm sorry.
having
No, you got
48 chromosomes. All right, so the chromosome 2 fusion is based on the apes having 48 chromosomes and humans having 46. And according to the atheist, the chromosomes fuse when the common ancestor split millions of years ago. Okay, so if that's what the argument for chromosome 2 fusion is, then I'm going to go ahead and go out on a limb and say that this is them just trying to find something to fit their interpretation. I believe this makes a little bit more sense to me now because they say that they see a fusion inside of the chromosome. I'll research into that a little bit more, but I can understand why they would make that argument now. However, I feel like that's a pretty big stretch for going to the extent of saying, oh, that proves apes and humans have a common, or apes and humans are related. That's a pretty far stretch to take. Chromosomes are pretty important to our DNA. So I would have to really look into that and see what kind of arguments they have for that, because that sounds like a pretty wild claim.
Absolutely. It could have just as equally as well have been designed that way. When they offer these pieces of evidence and it's so quickly accepted, the honest way to go about it, I think, is to examine the claim, number one, that because apes and humans are related, so therefore is what accounts for this. Well, let's examine other species. For example, the wild hog and the javelina. They are not able to reproduce. They are distinctly or close enough, but also different enough. Do they share any fused chromosomes? I suspect it's not so parsimonious as they are offering. And like Ken Hovind said, right. Just because you see something that shares a similarity, it could have been designed that way. And then they simply respond with the question, well, why would God design things in a way that could be interpreted as having common descent? Well, that's the interpretation they're superimposing on the facts.
Right. And that's a large stretch for them to take to make that interpretation as well. So, yeah, I think that's straining at a gnat, to say the least.
Absolutely.
Yeah. That's a very interesting argument, though. Yeah, that's a very interesting argument. Wow. Is that Dave or Dan?
Dan, hi.
Wow, I've been wanting to talk to you.
How you doing? Oh, great. Hey, so I actually just happened to be, I was listening to a different thing, and I saw this was live, and I just happened to catch this bit with the chromosome fusion, and I just popped in since you got space, and if it's okay, if I can just kind of elaborate on that argument a little more, because it's a little more complicated than what everyone just said. Would that be all right?
Yeah, I haven't done too much research on it, so whatever you can add to this, I will be glad to look into.
Oh yeah, I'd be happy to. So the idea here is the way we ought to be thinking about this is all about hypotheses. You make predictions based on the hypotheses, then you see if the predictions are correct or not, right? That's basically hypothesis testing in a nutshell. So what the hypothesis is here, the underlying hypothesis, is that Homo sapiens are descended from some common ancestor apes that we share with chimps, and then if you go back further, with gorillas and orangutans and so on. So we look at all the other great apes. You look at the orangutans, chimps, gorillas, and they all have 48 chromosomes, 24 pairs. But if you look at Homo sapiens, we only have 46. We have 23 pairs. So what's going on there? If we are descended from them, then there needs to be, at some point, right, a reduction in number of chromosomes. You can't just like jettison chromosomes because you'll die. You need all your chromosomes. So the answer is that 2 of them got stuck together at some point, right? That's the hypothesis. So the prediction from that, very specific prediction, is that if you compare the human and the chimp and all the other great ape genomes, you should find all of the pairs of chromosomes match up except for one, where the humans are going to have a pair that's absent in the other ones. And the other ones are going to have 2 that are missing from the humans. But if you put those side by side, the 2 from like the chimps and the 1 from the humans, then they should match up together. And you could say, okay, these 2 fused into this 1 human chromosome. Does that make sense? That's the hypothesis, right?
Okay.
We should be able to
I have a
find
question though,
this
because
hallmark.
I have a question because there's something about that kind of gives me a red flag. If
Go
2
for it.
chromosomes fused, wouldn't that cause major problems with the whatever organism this is happening to?
So not necessarily. There's a lot of things that are pretty loosey-goosey about their number of chromosomes. Plants just like don't care. They can just double their whole genome and like they're fine. Polyploidy is a thing in them. An example where this you could have 2 things that have different numbers of chromosomes where they're basically all matching with each other. But the numbers are different is horses and donkeys. So when they hybridize, they get, depending on which sex is which parent, you get either a mule or a hinny. And those things are sterile because the 2 parents have different numbers of chromosomes, but they're healthy and they're perfectly viable. There are also lots of deer species where like things that are very closely related have wildly divergent numbers of chromosomes. So chromosome structure is actually something that's fairly variable. You could mess around with that without causing too many problems, which is surprising. But you know, that's what we see.
Okay. Yeah, that's an interesting theory that I'll look into because I haven't really had the time to dive into that.
So
yeah,
here's the thing. So we have here's the thing. So we have this prediction, right? We should find essentially a missing chromosome that humans have that chimps and gorillas and such don't. And that's what we find. So human chromosome 2, you can match up human chromosome 1, 3, 4, 5, 6, 7, all the way down to the X and Y, and you have an exact match in the chimps. But human chromosome 2, you can't find that in the chimp genome. But there are 2 smaller chromosomes in the chimp genome that you can't find in the human genome. And we could do this thing. This is an older genetic technology. It's not sequencing. It's called staining, where you treat DNA with a chemical, and different regions of the chromosome pick up different levels of the dye. So you get what's called a banding pattern, where parts of the chromosome are very light and parts of them are very dark. And it's an older technology from like before sequencing was a thing. And you can line up the human chromosome 2 and the 2 chimp chromosomes, and the banding pattern is exactly the same, right? They line up exactly. And then the really interesting point is that, so do you know what telomeres are in a chromosome structure?
Yeah, so I haven't started researching into any of this. So yeah, a lot of these words you're using are roughly new to me. So yeah.
Oh, good. So a telomere is just the end of a chromosome. Basically, chromosomes have protective caps on the end where, so if they were to degrade, you don't like eat into any of the actual useful gene sequences. You have little protective caps that if they break down, like it doesn't matter. They're called telomeres. And these are like very specific repeating DNA sequences. And so a very specific prediction of this fusion hypothesis is that in human chromosome 2, we should find at the purported fusion site, we should find those telomere repeats. And we do. You find telomeres, you have the whole chromosome, right? So you've got telomeres at one end, and then and then in the middle of the chromosome, you've got a degraded telomere. And then facing the opposite direction, you have another degraded telomere, and then you have the rest of the chromosome and a telomere at the other end. So you have these end-to-end telomeres that are degraded at the purported fusion site where those 2 ancestral chromosomes would line up, exactly as you would predict. Now, a little while ago, somebody, I don't know if it was you or somebody else, but they said like, and this is held up as proof of human-chimp common ancestry. It's not proof. But it's the best we can do in science, which is a confirmed prediction. It is a specific confirmed prediction. And if we couldn't find something like that, if we couldn't find a direct chromosomal fusion between chimps and humans, then that would be a big red flag against human-chimp common ancestry. But the fact that we do find it, again, it's not proof, but it's really strong evidence in favor of it.
Okay, so here's a question I have, though. You said that this was a confirmed prediction, right?
Mm-hmm.
Well, my question was, is it a prediction, or did they find this and then add their theory to it?
This is a confirmed prediction because we didn't actually make this. So we made this prediction when we were like humans. Okay, so you have to go back like over 100 years for human-chimp common ancestry. You have to go back to the 1800s from that. We didn't even know what DNA was then. So then once we figured out what chromosomes were, which was early 1900s, that became a thing with like Thomas Hunt Morgan. It was like early 1900s. We kind of figured out that chromosomes have to do with inheritance. So at that point, you could look at the human genome and the chimp genome and see there are different numbers of chromosomes. So by the mid-20th century, we can say like, if humans and chimps share a common ancestor, there needs to be a chromosomal fusion. But we couldn't we didn't have the biotechnology to do the analysis to show whether or not that would be the case until way more recently than that. And specifically, the sequencing wasn't really a thing that we could do at that level until the 90s or more recently, because we didn't sequence the human genome until the early 2000s. So like in terms of like, is there a telomere sequence in the middle, right? This little end-to-end telomere sequence, we couldn't show that was there. I forget when the chimp genome was finally sequenced, but it was more recently than the human. And the human was like 2001, I think. So, these are predictions that go back decades at least. And we didn't actually have the biotechnology to test them until very recently. And they're accurate predictions.
Okay, yeah, I'm definitely gonna do some you definitely helped me understand y'all's argument a lot more. So now when I go to research this, I'll actually know what I'm looking for.
Yeah, just when you research it, watch out for. So one thing that you'll run into when you research it is that creationists will say that there's actually an active gene that spans the purported fusion site, and therefore it can't be a fusion site because you can't have an active gene across a fusion site, which would make sense if that was actually an active gene, but it's not. It's a pseudo gene, so it's a degraded inactive gene, and it's a member of a family of pseudogenes that only occur at telomeric sequences, which is to say this family of pseudogenes only exists. There's a bunch of them throughout the genome, but every other instance of it only occurs immediately adjacent to telomeres in a region called the subtelomere region. So it's like you've got your telomere, and then behind that, you've got the subtelomere. Every other instance of this pseudogene occurs in subtelomere regions, except for the one that exists in the middle of human chromosome 2, next to the degraded fusion site where there are telomeres. So just watch out for that. It's a common creationist argument that you find, but geneticists will laugh at you if you make that argument because it's really bad.
Okay, yeah, I'll definitely look. I'm almost done with my presentation that I'm working on for Chlamydomonas. So once I finish that up, I think that's the next one I'm going to go look into.
Oh, I'd love to hear. This is the Heron thing, the Heron 2019, the de novo multicellularity and Chlamydomonas thing?
Well, the one that I've been focusing on with Taylor, which is Snake Was Right. It's the life cycles of Unity to multicellular. That's kind of the one that he's been wanting to talk about. So that's
I think that's the earlier. Yeah, there's Chlamydomonas is that wonderful. That's so that's the green algae that never had multicellularity in its evolutionary history, but they induced the evolution of obligate multicellularity with predation. And then you take away the predator and it stays multicellular.
That's the other paper. That's the
classic,
paper that you're referring to. Yeah, the paper you're referring to with the predation, that's the one that me and Luke have been debating. The one that me and Taylor Snake Was Right has been debating is without the predation, with the centrifuge. So that's
the one by right where they separate out single versus unit. Yeah, right. So they use the centrifuge to select for the multicellular. Yep.
Yeah. Mm-hmm. Yeah, so I'm almost done with my presentation on that one. We kind of did a rough draft of that on Standing for Truth. If you want to go watch that whenever you have some time, I don't know how busy you are. But yeah, as soon as I finish my Chlamydomonas research, I think I'm going to make that my next topic to look into, because the way you explained it, I now understand what the argument is saying. So now I can go look at it and, I actually will understand what the claims are that they're making. So that'll definitely help with a good starting point.
Yeah, check it out, but just don't fall for the gene spanning the fusion site trick, because I'm not joking when I say geneticists will laugh at you if you make that argument. It's a telomere associated pseudogene. So it's actually predicted that it would be there in the middle of the chromosome.
So, Dr. Dan, thank you very much for clarifying for that. I just wanted to have a brief interaction
Please.
with you on that. Standing for Truth will be in here shortly. I'm sure it'll be a handful on this topic, but just for my own benefit, you mentioned that that this was a testable prediction that favored on the side of evolution. Certainly, I can agree to that interpretation. However, hypothetically, if this fusion chromosome that was discovered had not been the case, in other words, if it had been the case that they found an intact chromosome without the evidence of the degraded telomeres embedded in the middle, can you think of a naturalistic way in which to still account for that?
Can I think of a naturalistic way to account for a chromosome that is present in the human genome,
Right,
not in the chimp genome, with no telomeres in the middle?
right. Can such data still be interpreted by means of natural selection, beneficial mutations, in which evolution would still be upheld in the end?
So I do want to point out that I'm not sure natural selection is responsible for a lot of this. Chromosome fusions tend to be pretty probabilistic and in many cases without strong fitness effects. So I don't know that natural selection necessarily needs to be invoked here. And probably the degradation of the telomeres in the middle, they're not big, right? Telomeres have a typical length. The ones in the middle of the human chromosome 2 are a lot smaller. They're degraded. That's not due to natural selection. That's due to the removal of purifying selection. So what once was evolutionarily constrained is no longer. So it's not, just want to clarify that it's not necessarily selection that's like necessarily the thing driving what we actually see in the genome there. There's a lot of it's going to be neutral processes or non-selective processes.
Okay, that's fair.
So I think in this hypothetical, it would depend on the structure and the sequence of the chromosome, because what we know with both structural analysis and sequencing is that human chromosome 2 is a strongly matches the two missing chimp chromosomes, right? So you have these two smaller chimp chromosomes and they're not present in the human genome, but you line the sequences up next to human chromosome 2, then like it matches. If that wasn't the case, then it wouldn't be a confirmed prediction, because what we see is what we predicted. So in this hypothetical where the sequences of the chromosomes that don't match between the two genomes, in this hypothetical where the sequences are very different, then that would not confirm the prediction. Yeah.
Indeed, that would be true. And just as a layman coming at this, I just want to offer this quickly, and then I'll let Donnie take over, is that because God is said to have created man last on the sixth day, I see, or was it the sixth or fifth day? I forget. I think it was the fifth day. Sixth day, he rested on the seventh. That I see it as God working with a creation, a genome, a progenitor genome from which he tinkered here and there to create the different kinds, in which case between the chimpanzee and man, it was because of their overall similarity that is what accounts for the same similarity that we see in the genome, in this case the chromosomes, and that the fusion was, I would take that as evidence for an act of divine creation. I know that may seem as a weak proposition to you, and I can't blame you for that. This is something I'm going to have to look into further. because even in the Bible it says that God created Adam out of the dust of the earth. So it would beg the question of whether or not he's using brand new dust, or exactly what kind of workbench he was working with. So that's a fair point. So thanks again, Dr. Dan, for giving me that clarification. I'll let Donnie take over.
Happy to.
Dan the man, long time no
Donnie,
see, long time no.
It's been a while. It's been some time.
It's been some time. I hope you've missed me as much as I've missed you. How you been?
My heart aches for how long it's been. Let me tell you.
Well, that's why I joined. I sense the sadness.
No,
in
let's
you, Dan,
let's talk about Chromosome 2. What you got? Bring it.
Well, some interesting points, some good points, especially with the confirmed prediction aspect of the fusion. And so where I could start, I think, is if we were to assume, for sake of argument, Praise, if you could share my screen real quick.
Oh, here we go,
Dan
dude. It's already midnight
appreciates
for me. I don't have
visuals.
time for all the slides. Just say it. Just use
We'll be
words.
quick. We'll be quick.
Oh, it better be,
But
man.
this
'cause I
stuff
gotta go
is
to bed soon.
technical.
No,
No,
it's
it's supposed to be.
Donnie, can I just say one
wait,
time, we got to chat about like graphic design, because this blue background with the black text and the tiny
rough
little
on the
font,
eyes.
it causes me physical pain.
It's just more evidence of genomic degeneration. Dan, you messaged me what color you prefer, and for our next talk, when you have more time, we could do it on your channel.
Yeah, that'll be 2020.
I will personally alter the color of my slides to your liking. So I would say, even if there was a fusion event, I'm not totally against there being a fusion event, because we can go slide by slide, point by point. I think there's strong evidence that there's not a fusion event. Dan, you pointed out that you believe it's a weak line of argumentation to say that the fusion site itself represents a functional component of the DDX11L2 gene. We can touch on that a little bit.
It's a pseudogene. It's a pseudogene. It's not a gene.
Well, I definitely have some thoughts on that, but let's start here. So even if there was a fusion, I'm not totally against it, because as you can see here on the slide, it would be an apomorphy, since it is a trait observed in a single lineage. So basically the fusion, correct me if I'm wrong, Dan, from your model, from your position, the fusion would have taken place in the human line sometime after the split, right? Humans and chimpanzees go back to a common ancestor 6 to 10 million years ago. At some point in the human lineage, there would have been a fusion event. Is that right?
Yeah. So on the hominin side of that divergence, there would have been a fusion on the human lineage side, not the chimp lineage side. It is late, so please forgive me for asking, or maybe requesting. Can you be quick to get to the point here?
Well, that would be my point. You
so
just
the point is what? That it's an apomorphy? That's just a derived trait. Like, what's the... Yes, it's derived specifically in the hominin lineage.
Right.
Okay.
And so if there was a fusion, the biblical model, we have what? We have a flood bottleneck where you have 8 people. We actually have another bottleneck with the Tower of Babel. And so there could have easily been a fusion within that time period where genetic drift can allow for this discrepancy, this change in the genome to become fixed. So either way, I would say it's agnostic to the debate of creation versus evolution, since, as you've admitted, it is something that would have happened in the human line. And so that could either be evidence for evolution or creation. Go ahead.
Can I make sure I understand what you're saying? So you're saying that humans would have been created with 48 chromosomes like the other great apes, and then at some point, due to whatever genetic factors, the fall, small population size, drift, bottlenecks, whatever, that this fusion could have occurred within the young Earth human lineage, and all extant Homo sapiens now have 46 due to this end-to-end fusion. Is that that's the possibility that you're proposing here, right?
That is a possibility.
Okay.
Humans.
So yeah, got it. So here's why that's wrong, to not to put too fine a point on it, because what I'm saying is that this is a specific prediction of evolutionary theory, right? Human chimp common ancestry We can observe the different numbers of chromosomes, the different genome structures, the banding patterns in the different chromosomes, and we make a specific prediction that we should find a unrepresented chromosome in Homo sapiens and 2 unrepresented chromosomes, right, that don't have a counterpart in the chimp genome. And those things, when you line them up, should have a certain set of features. They should have sequence homology. They should have end-to-end telomeres. There should be a vestigial centromere in human chromosome 2, right? Those are specific predictions that must be the case if the evolution model is correct. Now, you're saying that the creation model can accommodate those observations, the observations that we find that those predictions are accurate. And that's true. The creation model can accommodate those observations, but that's a different thing from saying that they are necessary predictions to be
Dan, let me address those points
with the creation model.
Okay, I appreciate that. I also heard some background noise, whoever that's from. Since it's Dan and I interacting, please mute. Okay, so I would argue, going back to your admission, obviously this would have been a fusion event that occurred in the human line. When we actually
it's an admission, that's like the point.
But well, here's the thing: you want to use this as evidence for
a relationship between
humans and chimpanzees.
Yes, because it's a confirmed prediction. Yes.
Now you're saying it's a confirmed prediction, and I am not necessarily arguing against that claim. But what paper, what year, who made the prediction? How was it predicted? What was said? Was it super specific? Was it a little vague? Do you have the answer to that?
I do about an hour-long human evolution lecture every summer, and you're welcome to come on sometime and we can walk through it. But I mean, suffice it to say, yes, this was incredibly specific prediction.
Who made the prediction? What's the name of?
I don't remember the specific paper. It's been a while. Like this, and when I say it's been a while, I mean this was something from decades ago. This was a long time ago. This was a very specific human prediction.
Okay, so
it was from before sequencing. It was from before sequencing. The prediction was from before sequencing.
Okay, so you're saying before we knew that humans have 46 chromosomes and the great apes have 48, they made this prediction and it was specific.
So the prediction is based on the fact. So did you catch my conversation earlier with Jamie? Because what it was is go back over 100 years, go back to the 1800s and we say human chimp common ancestry. Then you get to the early 1900s. And that's where we figure out chromosomes are a thing for heredity. It wasn't until the early 1900s that people knew that chromosomes mattered for heredity. Right. So you get to the early 1900s, and now people are looking at different genomes, and you could stain the cells and count the chromosomes. So it's somewhere between, I don't know specifically when, but it was somewhere between 1903 and 1940, probably, give or take, that we realized chimps are 48, humans are 46. It's probably somewhere in that range. And so now we can make specific predictions, right?
Okay, so it was predicted. So it was predicted after the knowledge was accumulated that humans have 46. Okay.
Yeah.
And that's fine. I just wanted an answer to that. I don't want to spend too much time on one point because I know you don't have a lot of time. I would argue actually when you do compare chromosomes in humans and in chimpanzees, I don't want to spend too much time on this because I do want to argue for the functional nature of the so-called fusion site, which I know you disagree with the most. But I would argue, for example, I'm sure you're familiar with the Ventura paper. I know you are. I've seen you guys critique it before. And so there are aspects of human chromosomes and your great ape chromosomes, specifically chimpanzee and gorilla chromosomes, that do have some pretty significant differences. Here in this paper, it talks about the presence of these large blocks of subterminal heterochromatin and this lack of satellite DNA around human chromosomes.
Yeah. So what's heterochromatin specifically?
It's repeated DNA basically.
Not necessarily. It doesn't have to be repeated, but structurally, what's heterochromatin?
Structurally, what is heterochromatin?
What is heterochromatin?
Well, it's basically pieces of DNA within the chromosomes that is repeated oftentimes. That's my understanding.
It doesn't have to be repeated though. So the characterization heterochromatin is independent of the sequence. It refers to the physical conformation of the DNA.
Okay.
What does that mean? What is heterochromatin?
Well,
Are you making an
I
argument
think
that
you're
you
playing
don't-
word games here
making an
because
argument where
it
you
is.
don't know what the words
Just wait. Dan. I want you to answer the question. Why
I
is
will.
this DNA? Well, why is this structure
Yeah.
within the DNA? These structures within the
I'm
DNA.
answering. I'm in the process of answering that question, but I think you're making the argument without knowing what the words mean. Do
Okay,
you know what
how would you
heterochromatin
define
is
it if
structurally?
it's not just repeated pieces of DNA
I
basically
want to. So I just want
around
to
the
be clear, though.
chromosome? That's my
You
understanding.
don't
So
know.
if you want to,
Okay, so you're wrong. Okay, so heterochromatin refers structurally to DNA that is in a condensed state in the interphase nucleus, right? So do you know what those words mean?
phase in the
Condensed, so like structurally it is supercoiled rather than
Okay.
being in a diffuse state, right? So the nucleosomes are not exposed, right? They're supercoiled so that you don't have like access to enzymes that would do things like express genes. Okay, that's what heterochromatin is.
Well, it's thought to be tandem repeats.
It
Am I wrong?
is largely repeats, but it doesn't have to be because
and
it's cell
here's the
type
thing.
specific.
Listen, here's the thing. You just want to posture because I did give the correct answer, and then you want to get hyper technical, which I'm
You
fine
didn't
with.
give the correct answer.
No. I gave the. I've seen you all talk about heterochromatin before. I've studied heterochromatin.
Mm-hmm.
It's primarily thought to be tandem repeats, and it's condensed pieces of DNA known as satellite DNA. Okay, fine. Answer the question.
Yeah.
How come the presence of this is lacking around human chromosomes, where it shouldn't be lacking if we do share a relationship, or specifically if there was a chromosome 2 fusion?
So if you have highly condensed DNA, it's not being expressed. It's not being used. So that DNA is not under purifying selection. So deletions or translocations or rearrangements in those regions are not selected against. That's why. So you tend to have structural differences in regions where you have heterochromatin in one genome, but it's missing in closely related genomes. Because
is it missing because it was lost over time? It was lost over time?
Deletions are relatively common. If they affect a gene, then that tends to be highly deleterious. But if it affects exclusively heterochromatin,
so at what point in the split between the humans and chimpanzees' common ancestor was all of this satellite DNA, this specific kind of DNA, lost? At what point was it lost?
So we can't actually say that it was gained or lost specifically if we're only looking at chimps and humans. And I'm not familiar with the specific regions that you're talking about. Are you talking about regions in chromosome 2 that are shared across humans, chimps, gorillas, orangutans, and other apes? Or are you talking specifically about chimps and humans? Because that matters for the question of when was it lost.
I do believe it's species specific, but specific to chimpanzees, gorillas, and humans. The major differences are with those three. If it's
chimps, gorillas, and humans, then what probably happened is that it was a deletion that affected the hominin lineage, but not the other two. And
the hominin lineage, you're saying?
Hominin, so the
hominin,
human,
right? So the human line,
human side of the human chimp divergence, and so because it's heterochromatin.
I just want to ask you: is the function of the heterochromatin to make the DNA more condensed, like you were saying? Is that the function of it?
So the point is that heterochromatin is largely non-functional DNA, which, if I recall, I believe you don't think exists very much. But so the point of heterochromatin is that it's persistently condensed DNA.
Okay.
That is not doing a whole bunch of anything. So basically, they put it.
From my understanding, that is wrong, because I mean, I could go through papers where this kind of DNA does.
I'm just going to refer you to my chat with Fuzzrana from last year on that one. But not to get sidetracked on it. But so
would you deny the fact that there are papers going over the functional aspects?
Look, I don't want to get sidetracked from the human. I don't want to get sidetracked from the human chimp thing. So
let me just respond to what you were saying. Then I noticed that you said, well, probably there is a deletion, and see, what this is a lot of evolutionary philosophy coming from you, because you were saying we have this perfect match, exactly predicted humans and chimpanzees around these chromosomes, specifically
referring to specific regions.
But I am showing you that we have a specific kind of DNA that even those on your side are recognizing is a discrepancy. It lacks in human chromosomes, but it exists. It's in abundant, from my understanding, in chimpanzee and gorilla chromosomes. You're saying, well, probably some kind of deletion or something happened through purifying selection in the hominin line. Well, that's nice, Dan.
I
But
didn't
it's a
say it
lot
was
of storytelling. It's a lot of
purifying
evolutionary
selection.
philosophy.
With Stan, I'm not saying anything was selected here. I'm saying it was neutral. It's stuff that you don't need. So that's the
But that's
not
where your
selection,
evolutionary
though.
philosophy
I don't know where you're
comes into
getting
play. Okay,
selection.
I'm looking at a paper right now. It's called "Functional Significance of Satellite DNAs: Insights from Drosophila." So there are papers going over the functional aspects of satellite DNA and
no, I know they
heterochromatin.
exist.
Well, okay,
I know
okay.
they
I know you don't have a lot of time, so I do want to move on to. I would argue.
Well,
Okay,
hang
okay,
on.
go ahead.
Can I get a word in here, because I feel like you're jumping around and you're ignoring the actual point that I came in to make.
Well,
which
because
was
you're saying, Dan, you're saying more things wrong in one sentence than words that are in that sentence. You're saying a lot of storytelling on how this was lost. You're saying, well, it's just non-functional junk, and so it can be deleted or lost over time. That's wrong. That's false. It's not just non-functional junk. You're saying that, well, the DDX11L2 gene is a pseudogene. Again, that's evolutionary philosophy. Okay, go ahead.
So I would like everyone to pay attention here to what Standing is doing. I came in to answer a very specific question from Jamie, and I saw people were giving Jamie crap in the chat about this. I don't know why people are doing that. That was nice. Jamie, thank you. I think we had a nice conversation. Now, I brought up a couple very specific points in terms of the structure of the chimp chromosomes and a couple very specific features of human chromosome 2. And now Standing comes in and is talking about all these other things that aren't related to what I brought up.
It is related to the chromosome 2 fusion.
Please don't interrupt me. Please don't interrupt me. I am happy to have a conversation if you want to set up a time and we can talk about all this other stuff that you seem to want to bring up. But that's not what I came in here to talk about specifically.
It's all the same topic. It's all the same topic, and you're the one that started interrupting when this discussion started. I barely got 3 words in before you interrupted.
Fusion site? Do you have anything to say about the fusion site or the pseudogene, the head-to-head telomeres, the vestigial centromere? Do you have anything to say about that stuff?
Okay,
the confirmed predictions.
I just want to sum up this argument for the audience so they can see what we're talking about. I came in and told you, for sake of argument, if there was a fusion, I don't think it's a problem for the creation model because it is an apomorphy. It would have occurred in the human line.
What does apomorphy mean?
Well, if you look at the screen, a novel evolutionary trait.
Yeah, it's a derived trait, right?
To a particular so it's unique to the human line. But we cannot. But then you said, well, it doesn't matter because when we look at human chromosomes and chimpanzee chromosomes, it matches predictions and expectations. And I showed you one paper that says, no, it doesn't because of this lack of satellite DNA.
You're talking about two different things.
I do want to move on.
No. We're not going to move on from that because you're conflating two different things. The predictions that I'm talking about have nothing to do with satellite DNA. Well, you didn't even specify
I am arguing,
that satellite DNA was in
You're misrepresenting my position. You're misrepresenting my argument. I am saying it could have happened in the human line.
I agree.
To genetic drift, bottleneck, sometime after
the
flood.
It could have, but it's not a specific required prediction of your hypothesis. It is a specific required prediction for the evolutionary hypothesis, and it's a confirmed prediction.
No, but I would say there's a lack.
It's not equivocal.
I would say there's a lack of consistency with the evolutionist explanation because of the lack of satellite DNA. This very specific kind of DNA.
That is a different thing.
I would disagree with that. I would disagree with that. Okay, so we can move on, though, and we can agree to disagree, okay? So you're trying to say that it's a pseudogene. Well, firstly, anytime you see pseudogene, especially for the audience sake, that is the evolutionary assumptions, just like we saw with the satellite DNA.
No, it's functional. It's been excessive.
Dan, you have to admit, you're interrupting me, too. There's healthy interruptions. You can have as much time as you want to respond, okay? I understand you're going to disagree with a lot that I have to say. I disagree with a lot of what you have to say. I'll try not to interrupt anymore. You try not to interrupt anymore, okay? We understand that many pseudogenes are now necessary to sustain healthy life processes in the cell. I understand you disagree with that, and I appreciate you not interrupting, okay? So what we have here, and Dr. Jeffrey Tompkins, who I know you love so much, he says the purported fusion site is a functional DNA binding domain inside the first intron of the DDX11L2, or 11L2, non-coding RNA helicase gene. So he's saying basically this so-called fusion site, which is about 800 base pairs.
That word? Was it non-coding?
What does that mean? Non-coding DNA
Yeah, what does
has
that mean?
a lot of necessary essential functions.
What
And
is
actually,
it? What is it?
Non-coding DNA is not coding DNA, so it's
So,
not.
but you're saying
coding.
you're saying it's not a pseudogene. What does non-coding mean?
Well, when you say pseudogene, you're trying to say, well, this is a genetic mistake, a broken gene,
No.
an evolutionary leftover.
No. What's
Okay,
the definition of
what is it?
pseudogene? What's the definition of pseudogene? Do you know?
A pseudogene is a broken protein coding gene.
Right. And it's broken, so it's
but you're assuming that it's broken.
Gene.
You're assuming that it's broken. I am saying that we know regulatory regions can be non-coding, and so you're looking at this and you're assuming that it is a broken protein coding gene. I am saying no, it's not a broken protein coding gene. It's a wait. A lot of the beginnings of your sentences start in the middle of mine. I am saying that this non-coding RNA helicase gene, okay, the DDX11L2 gene that overlaps the so-called fusion site. The so-called fusion site is acting as a second promoter for that gene that is not a broken gene. And here's the last thing I want to say is, how do you get highly essential and functional genes by slamming together two chromosomes? Go ahead, take your time.
So here's the thing. One, we know this is not a sequence. This region is not under purifying selection. The promoter that Tompkins is pointing out here, promoters are. Does everyone know what a promoter is? Does everyone know what a promoter is? Stanley, why don't you explain to everybody what a promoter is?
A promoter initiates transcription.
A promoter is a transcription initiation site. They are highly sequence specific. So in order for a promoter to work, it has to be sequence constrained. It has to be under purifying selection. Functional promoters, if they mutate, are selected against, right? The mutated non-functional ones are selected against. This region, this fusion site, is highly degraded and it's not experiencing purifying selection. So it's not a functional promoter. What Tompkins is saying is, you look at this
what makes you think it's highly degraded? Unless you want to just take your time and I can respond.
I'm getting there.
Take your time.
So what Tompkins' argument is that you've got this pseudogene in the subtelomeric region, and there's an alternate start site on the far side of it. So the fusion site itself occurs within an intron of this pseudogene. Now, he calls it, in the quote on screen right now, a functional DNA binding domain inside the first intron of the non-coding RNA helicase gene. Right. It's okay. So that's a little bit misleading from Tompkins there, because there are protein encoding genes, and then there are RNA genes, right? There are genes that make RNAs, the RNAs do something. But a helicase is a type of protein, right? It's an enzyme that does a thing during DNA replication. If it is a pseudogene that is non-coding, it's not making a helicase, because a helicase is a polypeptide, right? And this sequence does not ever result in a polypeptide. So just right off the bat, what Tompkins is saying here is wrong. Like, he is conflating things that are not the same thing right there, right? What you had on screen.
Are you saying that he's just lying? He's wrong about this quote?
I don't want to say whether he's lying or whether he doesn't realize he's wrong, but it's not a protein coding gene, and it's a pseudogene for a helicase. It's a pseudogene.
Nobody's
for
saying
a helicase.
He's not saying it's a protein coding gene. And he points out here that the fusion site sequence binds to at least 12 different transcription factors, including RNA polymerase 2, a key enzyme that transcribes genes. Are you denying that? Are you saying that he's wrong about that?
No, RNA polymerases love DNA. They grab on all over the place. They are huge fans of DNA, and it's not hard to get RNA polymerases to bind to DNA. It's very easy to do. You can randomize promoter sequences and you still get transcription. You can literally just take 100 random nucleotides and you still get transcriptional activity. That's been experimentally shown. So, like, you got to show me if you want to show me this is a functional gene, you got to show me transcription, you got to show me translation, you got to show me purifying selection.
Okay, well, here he says,
you get out of this is transcription.
Well, there's a lot. I mean, I've got pages and pages of notes on. Is there
in all those pages, is there translation and purifying selection? I don't think so.
Why would they be so okay? So we have DNA to RNA to protein, transcription to translation, translating for a protein. Why would there be translating? It's non-coding. It's a
okay. So it's a
functional
pseudogene. So
non-coding RNA gene.
Great, done. Great. So there's no translation. Is there purifying?
Well, why
selection?
would there be translations? It's an active promoter. Notice here the DDX 11L2 long non-coding RNA gene.
I'm not saying.
I just want to finish my sentence, Dad.
I'm not
I
disagreeing.
didn't interrupt you. Okay, this gene, long non-coding RNA gene. You said it's not functional at all. You said it's a pseudo gene. It's a broken protein coding gene. Well, notice this RNA gene containing.
Yeah, it's not protein coding.
Well, it's a broken which makes it non-coding, and it's
okay,
degenerative coding.
It's not. It's no longer protein coding.
It's no longer protein coding, but at one point it was. RNA gene containing the alleged fusion sequence encodes several transcript variants expressed in at least 255 different cell or tissue types. I mean, that's pretty significant, is it not, Dan?
No, I mean, honestly, no. To be frank, no.
It's doing something. It's doing something.
Yeah, transcription. I don't know if people appreciate just how messy transcription is. I don't know if that's like a common thing that people think about, is that transcription is a noisy, messy process. And even when it's highly regulated, which it largely is, there's still a lot of messiness in terms of what enzymes can grab onto.
It sounds like you're assuming it's messy. It seems like more
It's not an assumption. It's been evolutionary documented that it's messy. This is not an assumption, sir.
I mean, does it not require a lot of energy within the cell to add a nucleotide, a single nucleotide to a growing
adding a single nucleotide requires 1 ATP or equivalent. Yes.
It still requires ATP synthase. That's 1 nucleotide to a growing RNA chain. I'm just seeing a lot of evolutionary philosophy, a lot of evolutionary based assumptions with very little evidence. Again, we see that it's doing something. We see that the site, as you've admitted, is very degenerate. It's only about 800 sequences long. Wait, I'm not done.
Or is it functional?
Go ahead.
Is it
it's functional because the so-called fusion site is a promoter for this long non-coding RNA helicase gene that actually overlaps it. It overlaps the fusion site.
Yeah, Tompkins' argument is that the fusion site is that the promoter is on the far side of it. That the first exon is on the far side from the rest of the pseudogene. That's Tompkins' specific thing. Is that you have a start site that spans it, right, and that the fusion site occurs within an intron.
Right. Yeah, inside the first intron of the DDX. 11L2 non-coding RNA helicase gene. And so it does. It spans the fusion site. And so and you admitted earlier with Jamie that yes, if this was a functional component of the gene that spans it, then that would be good evidence. Sure, maybe this wasn't the result of a hypothetical fusion at some point in the hominin line. And that's what we're seeing. We are seeing a lot of function associated with it.
We're getting bogged down in the minutia here, and I want to make clear for everybody what the point is before I go to bed, because I am going to go to bed in a couple of minutes. But I want to make very clear what the point is here. When we first took a look at the human and chimp genomes, what people expected to see was the same number of chromosomes. That was the original idea, right?
Right.
That was the original prediction that,
and it failed. It was a failed prediction.
And it was wrong, right? That was wrong. That was an incorrect prediction. So what we said is, okay, if it's the case that humans and chimps share common ancestry, then there has to have been a fusion in the human side of that lineage descended from our common ancestor with chimps. Right, there must have been a chromosomal fusion.
But there wasn't.
If that is the case, if that is the case, this is the confirmed prediction. If that is the case, then we need to find certain features. We need to find an end-to-end telomere in the middle. We need to find a vestigial centromere somewhere in the middle. And
you don't find any of the two.
And these features need to match up in the human chromosome and the 2 chimp chromosomes. Now, when you sequence those chromosomes, you find the banding patterns. Hang on one second, and I'll show you a picture, because you're saying they don't match up, and I'm going to find a picture.
Well, firstly, we've already shown many discrepancies.
Oh, hang on, no. You said they don't match up, and I'm going to find a picture.
Yeah, but here's the thing. You've talked about the telomeric repeats. You've talked about the cryptic centromere. Let's go one at a time, one topic at a time. Notice here on this slide.
Here's the picture I'm looking for. I'm going to show this one picture, because again, I need to go to bed. I want to address.
Are you talking about synteny? Wait. Are you talking about synteny?
Hang on. Just give me a moment.
Well, as you're finding it, I want to
my
screen. If you got to go to bed, then there's a lot of slapping and running
here. Here we go. Here, can I just can someone just put my screen on just for a second? It's literally just, it'll only take a second to show this because Standing is saying they don't match. And like, this is the banding pattern. So this isn't the sequence similarity. This is just when you stain them and it's looking at different features of the structures. They do match. You can look at this. Like they match.
Well,
okay,
you're going to find a lot of similarities, sure, when you're looking.
So that's okay. Well, that was a lot of discrepancies
too.
Well, my point is, this was a specific confirmed prediction that the structures are going to match. They're going to have specific features in the same places, which we find. And when you look at the sequences between them, the sequences are going to match. You find the same gene sequences in the same places on human chromosome 2 and the 2 smaller chimp chromosomes. These are all specific confirmed predictions that must be true.
Wait, is this synteny or no?
Well, hang on. Let me finish my point, and then you could say whatever because I'm going to go to bed. But these are
I want
things...
you to stay while I refute your points, though. I want to get a concluding statement. So go ahead, finish, because I don't want you running.
These are confirmed predictions, and again, I'll summarize them, and I'd ask that you just let me summarize it without interrupting. So the size of the chromosomes, the banding pattern of the chromosomes, the structural features like telomeres and centromeres of these chromosomes, and once we had the capacity to sequence them, these specific gene sequences, finding the same genes in the same places, right? So we can line up these chromosomes side by side, and we find a bunch of specific features that are the same between the two, the human chromosome 2 and the 2 smaller chimp chromosomes. My big point here, and the sole reason I joined this stream to just make this point, is that it's not just that we said, oh, there's fewer chromosomes, must have been fusion somewhere. It's that we have these specific predictions that must be the case if human-chimp common ancestry is true. Those predictions, being accurate, don't invalidate the creation model, but it doesn't matter one way or the other. The creation model can accommodate it either way. But for the evolution model, you must have these similarities between these chromosomes. And it's exactly what we find. They are accurate predictions. And as I said at the beginning, I think before you were here Standing, it's not proof by any stretch of the imagination that of human chimp common ancestry. Science doesn't deal in proof. Math deals in proofs. Biology doesn't, right? It's not proof, but it's a series of extremely specific predictions that were very specifically confirmed. And that's why it's strong evidence of human chimp common ancestry.
Okay, Dan, I wrote down your points. I am going to address it uninterrupted, and then you know what?
Can I ask a question about that photo?
No. Jamie, it's me and Dan going at it. Then I want to give Dan the last word. Okay, I appreciate.
Hold up. I want to take in one question before he goes.
Okay, go.
All right, Mr. Dodgeball Dan.
You know, if we're gonna get into that, no, I want to finish my thought.
No, if you're gonna do that, like I'm sitting here for 45 minutes.
Are you scared of that question, Dodgeball Dan? Are you scared of that question?
Bubblegum, before
we're not gonna do this.
Lost,
you're scared of that question, or are you?
No. I'm gonna finish up with Standing, and if Jamie wants to say anything, I came in to talk to Jamie. Standing joined. We've been having a nice chat. If you're gonna come in and insult me, then I'm gonna leave. Like I got better things to do with my life.
Okay,
that's just the question.
Hey, Olive, please mute Bubblegum. I really want to get these points in because there's a lot of points on the table. If I can share my screen, and out of respect, I do appreciate Dan's time here and staying cordial for the most part. I'll give him the last word. Okay, so firstly, yes, they had to invoke a fusion when they realized the chromosome numbers didn't match. Otherwise, evolution fails. And when it comes to the DDX11L2 gene that does span the fusion site, it produces RNA that actually helps regulate the DDX11L gene family, as has been shown with other functional pseudogenes. And so again, the question that I asked, and I don't feel like was answered, but we could have another discussion on this later, is how do you get highly essential and functional genes by slamming together 2 chromosomes? Sounds unrealistic, sounds far-fetched, okay? When it comes to the region, yes, we should have found these telomeres. And what do we find here? Well, Dr. Tompkins, who also addressed this synteny argument in detail in his paper, I actually have the paper printed out. I'd recommend reading it. And he says, actual synteny between human and chimpanzee remains to be resolved until an unbiased assembly, unresolved, until an unbiased assembly of the chimpanzee genome is produced. When you look here, he says an end-to-end fusion as proposed by evolutionists would give a signature of at least 10,000 bases in length. But what we actually find is only 798 bases in length. The evolutionists would just say, well, there's been a split, and so a lot of those bases or sequences have been lost. But it turns out that when you actually do a genome search in all the different chromosomes of these telomeric-like sequences, you find them throughout the entire genome in all chromosomes, with a lot of them associated with significant function, such as gene expression. Okay. And if you read his paper here, Debunking the Debunkers, which I don't see the critics actually addressing, he points out in summary, chromosome fusions would not be expected to form complexly transcribed and regulated multi-exon alternatively spliced functional gene. This is a functional gene. And in this paper specifically, he refutes Ken Miller's arguments against what he's saying. And these are the arguments basically that are just repeated by people like Dan and other critics. And so I'd highly recommend that they get up to date on the evidence. So no, what they actually find is not what was predicted with the telomeres, with the centromere. Obviously, we see a lack of satellite DNA, and I just don't think it's looking very good for the fusion hypothesis. Last thing I want to say, when you hear the evolutionists like Dan talk about non-functionality or the genome's really messy or pseudogenes, assuming that a lot of these non-coding genes were once functional, unbroken protein coding genes, that is riddled with evolutionary philosophy and evolutionary bias, because we understand a lot of these pseudogenes are necessary to sustain healthy life processes in the cell. And so either way, if there was a fusion, the creation model can explain it. But what the data suggests is there probably wasn't a fusion because of the functional nature of the site itself. I yield, and Dan can have the last word.
Thank you. So I'll just run through a couple points really fast, and then Jamie, if you want to get anything just at the end, feel free. So one thing, the regulating the DDX11 family, I want to just refer everyone to, I chatted with Dr. Fuzrana from Reasons to Believe last summer, and we talked about this exact thing for a while. And the idea is that regulation is messy, and the way transcripts interact with, RNA transcripts interact with other RNAs is very messy. And we have very strong evidence that they will interact and interfere with each other, but we don't have strong evidence that it is actually regulation that is selected for. And again, I went into a lot of detail with this with Dr. Rana, and it would be a lot to kind of rehash all of that here, but I'll refer to everybody. You can find my channel, shameless plug, but I chatted with Rana, and we talked about this exact thing for like 20 minutes. Check that out. It was a really good conversation. We go into it. In terms of functional DNA from a fusion, I mean, fusions are really common. So, for example, I apologize. I'm gonna share my screen just one more time, and it's a figure of chromosomes in two different species of there are these deer called muntjacs, and like by any model, evolution, creation, old Earth creation, young Earth creation, by everybody's model, we all agree that these things share common ancestry. They're sister species. The sequences of all of this DNA are almost identical. What's different is the arrangement of the chromosomes. In one species, you have very few, very large chromosomes. In the other species, you have many very small chromosomes. The way you get from this to this is a whole bunch of chromosomal fusions, just telomere end-to-end fusions, right? So it's not like this is a hard thing to do. This happens all the time in evolution, in places where creationists and evolutionists agree
no, we don't.
that two things share a common ancestry. So that's on the end-to-end, how do you get all this information? Chromosome fusions are easy. It's not hard to do. In terms of the satellite DNA thing, satellite DNA is often species specific, largely because it's not under purifying selection. So what gets retained and what gets lost is there's not expected to be consistency across different lineages if they share a common ancestor with that specific DNA. Why are the telomeres so little? Because they're broken down. They're non-functional. They're not under purifying selection. So any deletions that affect them are not selected against. And then the last thing is just a complex transcribed multi-exon, whatever phrase it was of Tompkins. I want to make sure everyone's clear on this. transcription is really messy. And transcription costs energy, but so does regulation. Regulation is really, really costly. So you could actually compare the cost of a certain level of transcription versus the cost of completely eliminating that amount of transcription. And what you often find is that the cost of regulating it to zero is more than just letting it happen at a low level. And as evidence that this transcription that we're talking about is spurious is that it has multiple start sites. When you have functional transcripts that are either transcribed or not, it tends to have one start site. Sometimes you get alternate start sites and you get different structures. Largely, you have one start site and you need that specific thing. When you have multiple start sites that are close to each other, that's an indication that what you're transcribing isn't all that important because the sequence can vary a lot and it's not.
Well, Dan, that's kind of a long last word, man. That's not fair.
Last
word is like sum it up in a minute or two.
I'm going to give you the last word because I'm going to go to bed now, and you could say whatever you want.
Whoa, don't run away, Dan. I got one question.
No. So stop. I'm not going to. I'm going to give him the last word, even though it was a long last word. Closing statement. A lot I want to respond to. You're still getting the last word. Share my screen. I'm not even going to say anything or elaborate on this.
You're welcome to do it.
But your point, Dan, on the spurious nature of the DDX11L2 gene. Again, it's been addressed. These are repeated talking points. You're getting the last word. I'm not going to go through this slide, but I just recommend people check this out. Debunking the debunkers. It's been addressed. This is just talking points that have already been debunked. So that's all I'll say. Look at that article.
Look, you can say, like I said, you can say whatever you want. I need to go to bed. But the big thing I wanted to emphasize for Jamie was the thing I talked about specific evolutionary predictions. So I appreciate you all letting me come in and giving me so much time. That was a lot of fun. And Jamie and Standing, thanks. I appreciate it. I'll talk to you all later. Bye.
Dr. Dan, appreciate it. And so I'll if Dan's gone, I don't know, I screen shared. I'll probably cut this out and make it a clip. Dr. Dan versus SFP or something. But I do want to say, and I'm going to leave this out of the clip because to be fair to Dan, we're going to give him the last word. Everything that he's saying about the noise, the spurious nature of the fusion site, it's all been dealt with. Right here, one specific excerpt from Dr. Tompkins just absolutely blows that out of the water. He's already addressed it. Okay. Their biological significance cannot be measured simply by comparing them to protein coding genes. An important detail that a lot of the critics like Kenneth Miller, for example, who did say if there was no fusion, then there was no evolution. But this goes back to what I was saying at the beginning too. It is an apomorphy. And so there are some details about this, okay, that the evolutionists want to latch onto, like the repeats at the fusion site, for example. They're in forwards and backwards, okay? Nonetheless, it is an apomorphy. It would have happened in the human line. We can explain that according to our model. Even if there is a fusion, it's no help to them, okay? But there is some good evidence, like the functional nature of the DDX11L2 gene, that suggests there may not have been a fusion. So there's points on both sides. I'd recommend creationists.
So that was my conversation on human chromosome 2. I hope you all found that informative and entertaining. Thank you all for watching. Before you go, please remember to hit the like button, leave the comment, subscribe, all the algorithm-friendly things. Thank you again for watching. As always, don't get fooled.